So the lac operon is an example of positive control. If there is lactose present, the lactose needs to be digested, and therefore the operon will be switched on. Makes sense. There's also negative control. Okay? In other words, when something needs to be produced, we can use a negative control type of operon. Okay? That's going to allow that material to be produced until there's plenty of it present. Once there's plenty of the material, the full end product produced, then we can turn off that operon. The trip operon is also in E. coli, and it is a step-by-step chemical pathway to produce the amino acid tryptophan. This is an operator. Okay, it's an operon rather. Okay, one single promoter. You've got an operator, and that promoter and operator then switch on or off the genes that it controls. Let's take a look at a diagram. So here we have the trip operon, a regulatory gene upstream from the genes of the operon, produces an inactive repressor. So this is a protein that's going to be allosterically controlled. It's not in the proper shape to fit in the operator. Okay, so it cannot, as it's originally produced, it cannot turn this operon off. The promoter and the operator together work at the start of this operon. So without an active repressor, RNA polymerase can bind to this promoter site and move downstream, producing transcripts of all 5 of the trip operon enzymes. Those 5 enzymes are needed to make tryptophan from the raw materials in the cell. But Mother Nature has no time to waste. So if there's plenty of tryptophan, she's going to turn this whole system off. So tryptophan actually acts as a co-repressor. Tryptophan would fit here, changing the shape of the configuration of this protein, allosteric control of this protein. So we have plenty of tryptophan. Tryptophan binds to the repressor, changing the shape of it. That repressor now fits in the operator, okay, turning this whole thing off. Because if we have plenty of tryptophan, there's no need to produce the machinery needed to make more tryptophan. So if the trip operon is a negative example, then what we're saying is that ample tryptophan will stop production of more tryptophan. There's no need to make it. There's just no need to make it. Okay. If there's a lack of tryptophan, okay, so we can't, tryptophan being an amino acid would be necessary for protein production. If there's a lack of tryptophan, this trip operon will then be turned back on. Okay. Tryptophan as a co-repressor would no longer be causing the shape of that, okay, no longer be causing the shape of this thing to be changed. So here we're talking about changing the shape of that trip repressor, okay, by not being there. Okay. So the lac operon is a positive control. The trip operon is a negative control.